Hello there, and welcome to the Sleepy Library. Tonight, we are drifting into the microscopic realm of DNA, where the blueprint of all life exists in a quiet spiral of molecules smaller than you could ever imagine. This journey isn't about complex science or hard-to-grasp concepts. It's about wonder, about the gentle patterns that exist in every cell of your body, about the hidden architecture that makes you. Let your breathing slow. Let your mind rest. Tonight, we'll explore the molecular poetry written in every living thing, from the smallest bacteria to the tallest tree, from the depths of the ocean to the person lying next to you. These aren't just facts. They're invitations to marvel at the elegant simplicity underlying all the complexity of life. If you enjoy these quiet journeys, I invite you to like, subscribe, or share a thought below. It helps others find their way here too, one sleepy soul at a time. But for now, just breathe. Let the night settle around you and allow your mind to drift. Let's begin. DNA was discovered long before anyone understood what it did. In the year 1869, a young Swiss physician named Friedrich Miescher was working in a laboratory in Germany, studying white blood cells from discarded surgical bandages. He wasn't looking for the secret of life. He was simply curious about what cells were made of. Deep within the nucleus of these cells, Miescher found a substance unlike any protein or fat known at the time. It was rich in phosphorus and slightly acidic. He called it nuclein because he had found it in the nucleus. For decades, nuclein remained a mystery, a chemical curiosity without a clear purpose. Scientists knew it existed. They could isolate it and study its properties. But they had no idea that this strange substance held the instructions for building every living organism on Earth. Miescher's discovery sat quietly in scientific journals, while the world moved on, unaware that the key to heredity, to evolution, to the very nature of life itself, had been found lying hidden in the discarded remnants of human wounds. The shape of DNA is a double helix, but it took nearly a century to discover this. Throughout the early 20th century, scientists slowly pieced together clues about the nature of heredity. They knew that something passed from parent to offspring, carrying traits like eye color and height. They knew chromosomes were involved, but the molecular mechanism remained elusive. In 1953, everything changed. James Watson and Francis Crick, working at Cambridge University, proposed the structure of DNA based on X-ray crystallography images captured by Rosalind Franklin and Maurice Wilkins. The structure was elegant, almost impossibly simple. Two strands of molecules wound around each other in a spiral, like a twisted ladder. The rungs of this ladder were made of 4 chemical bases that paired together in specific ways. Adenine always bonded with thymine. Guanine always bonded with cytosine. This pairing wasn't random. It was dictated by the shapes of the molecules themselves, by the precise arrangement of atoms that allowed hydrogen bonds to form between complementary bases. The double helix wasn't just a shape. It was a solution to a problem. How does a molecule store information and also copy itself? The answer lay in the pairing rules. If you knew one strand, you could predict the other. If you separated the strands, each could serve as a template for building a new partner. It was a mechanism for both preservation and replication, encoded in the very structure of the molecule. DNA is incredibly long, but impossibly thin. In every human cell, there are approximately 3 billion base pairs of DNA. If you were to stretch out all the DNA from a single cell and lay it end to end, it would measure about 6 feet long. Yet this entire length is packed into a nucleus so small that you'd need a microscope to see it. The DNA strand itself is only about 2 nanometers wide. A nanometer is 1/1000th of a meter. To put this in perspective, a human hair is about 80,000 nanometers in diameter. DNA is tens of thousands of times thinner than a hair. This means that in your body right now, there are trillions of cells, each containing 6 feet of DNA, all coiled and folded with exquisite precision. If you could take all the DNA from all the cells in your body and stretch it out in one continuous strand, it would reach from Earth to the Sun and back more than 600 times. You carry within you enough DNA to span the solar system, yet it weighs almost nothing and occupies a space barely visible under the strongest magnification. This is the scale of life's architecture. Vast in length, minuscule in size, and perfectly organized in ways that allow a single fertilized egg to grow into a complete human being. DNA is made of just 4 chemical building blocks. Despite the incredible diversity of life on Earth, from bacteria to blue whales, from fungi to redwood trees, all living organisms use the same basic alphabet to write their genetic code. This alphabet has only 4 letters: adenine, thymine, guanine, and cytosine, abbreviated as A, T, G, and C. These 4 bases are all that's needed to encode the instructions for building every protein, every enzyme, every structural component of every organism that has ever existed. The simplicity is stunning. 4 chemicals, arranged in different sequences, contain all the information necessary to create the complexity of life. It's like writing every book ever published, every song ever composed, every thought ever conceived using only 4 symbols. The bases themselves are organic molecules made primarily of carbon, nitrogen, oxygen, and hydrogen. They're called bases because they're mildly alkaline in solution. Adenine and guanine are larger molecules with 2 rings of atoms, classified as purines. Thymine and cytosine are smaller, with only 1 ring, classified as pyrimidines. This size difference is crucial. A purine always pairs with a pyrimidine, ensuring that the width of the DNA helix remains constant throughout its length. It's a molecular handshake, a recognition between shapes that fit together perfectly, stabilized by hydrogen bonds that are strong enough to hold the structure together, but weak enough to separate when needed. The human genome contains about 20,000 genes. For a long time, scientists estimated that humans had around 100,000 genes. It seemed logical. Humans are complex organisms, so surely we needed more genes than simpler creatures. But when the Human Genome Project finally sequenced the entire human genome, the number turned out to be much lower than expected. Somewhere between 19,000 and 20,000 protein-coding genes. This was surprising. A simple roundworm has about 20,000 genes. Rice has more than 40,000. Humans, with all our complexity, with our brains capable of language, abstract thought, and art, have roughly the same number of genes as a microscopic worm. The answer to this puzzle lies not in the number of genes, but in how they're used. Human genes are more complex. They have more regulatory regions, more alternative splicing, more ways to create multiple proteins from a single gene. Complexity doesn't come from having more genes. It comes from using the genes you have in more sophisticated ways. Each gene is a set of instructions for building a specific protein or set of proteins. Proteins are the workhorses of the cell, the molecules that actually do things, build structures, transport materials, catalyze chemical reactions, send signals. Genes are the blueprints, proteins are the construction workers, and in between lies a vast regulatory network that determines when, where, and how much of each protein gets made. DNA doesn't just exist in the nucleus. While most of your DNA resides in the nucleus of your cells, packaged into chromosomes, there's a small amount of genetic material elsewhere. Your mitochondria, the tiny organelles that generate energy for your cells, have their own DNA. Mitochondrial DNA is circular, not linear like chromosomal DNA, and it's much smaller, containing only 37 genes compared to the 20,000 genes in the nucleus. These 37 genes are all related to energy production, encoding proteins and RNA molecules essential for the mitochondria's function as the cell's powerhouses. What makes mitochondrial DNA particularly interesting is how it's inherited. You inherit all of your mitochondrial DNA from your mother. Sperm cells have mitochondria, but they're left behind during fertilization. Only the egg's mitochondria make it into the embryo. This means that your mitochondrial genome is a direct maternal line, passed from mother to child without the genetic shuffling that occurs with nuclear DNA. Scientists have used this property to trace human migration patterns throughout history, following maternal lineages back tens of thousands of years. Your mitochondrial DNA connects you in an unbroken chain to your mother, your grandmother, your great-great-great-grandmother, and so on, back to the very origins of our species. Each cell typically contains hundreds or even thousands of mitochondria, and each mitochondrion has multiple copies of its genome, meaning that a single cell might have 1,000 to 10,000 copies of mitochondrial DNA. This redundancy is important. Mutations in mitochondrial DNA can lead to energy-related diseases, but having multiple copies provides a buffer, a genetic backup system that keeps cells functioning even when some copies are damaged. DNA replicates itself with astonishing accuracy. Every time a cell divides, it must copy its entire genome so that each daughter cell receives a complete set of genetic instructions. In humans, this means copying 3 billion base pairs, and it must do this with almost perfect precision. The error rate of DNA replication is incredibly low, about 1 mistake per billion base pairs copied. This level of accuracy is achieved through multiple mechanisms. DNA polymerase, the enzyme that copies DNA, has a built-in proofreading function. As it adds new nucleotides to the growing strand, it checks each one to make sure it's the right match. If it detects an error, it backs up, removes the incorrect nucleotide, and tries again. This proofreading reduces errors to about 1 in 10 million. But the cell doesn't stop there. After replication is complete, another set of enzymes scans the new DNA strands, looking for mismatches, places where the wrong base was inserted. This mismatch repair system catches errors that the polymerase missed, bringing the error rate down to 1 in a billion. Even with all these safeguards, mistakes still happen. On average, each time a human cell divides, it introduces 1 or 2 new mutations. Most of these are harmless. They occur in regions of DNA that don't code for anything important, or they change a codon in a way that still produces the same amino acid. But occasionally, a mutation occurs in a critical gene, altering a protein's function. This is the raw material of evolution, the source of genetic variation that natural selection can act upon. Without mutations, there would be no adaptation, no diversity, no evolution. But too many mutations would be catastrophic, breaking genes faster than selection could weed out the damage. DNA replication walks a tightrope between perfect fidelity and just enough error to allow change. The DNA in all your cells is almost identical, but not quite. When a fertilized egg divides, it creates 2 cells with identical DNA. Those cells divide again and again and again until there are trillions of cells making up your body. In theory, every cell should have exactly the same DNA. Perfect copies passed down through hundreds of generations of cell division. In practice, this isn't quite true. Small mutations accumulate over time. A cell in your liver might have a few mutations that a cell in your brain doesn't have. Most of these mutations don't matter. They're silent changes that don't affect how the cell functions. But in some cases, mutations in specific genes can cause a cell to behave abnormally. This is how cancer begins, not with a single mutation, but with an accumulation of mutations over time, each one pushing the cell a little further toward uncontrolled growth. Cancer is, at its core, a disease of DNA. There's another reason why your cells aren't all genetically identical. In certain specialized cells, DNA undergoes programmed rearrangement. Immune cells, for example, shuffle pieces of their DNA to generate millions of different antibodies, each one capable of recognizing a different foreign molecule. This genetic diversity within a single organism allows your immune system to respond to virtually any pathogen it encounters. These immune cells, with their rearranged DNA, are genetically unique, different from every other cell in your body. It's a kind of controlled mutation, a deliberate breaking and reforming of DNA to create variation where it's needed most. DNA is packaged using proteins called histones. DNA is astonishingly long, but it fits into the tiny nucleus of a cell through an elaborate packaging system. The first level of organization involves proteins called histones. DNA wraps around clusters of histone proteins like thread around a spool. Each spool, called a nucleosome, contains about 147 base pairs of DNA, wound around 8 histone proteins. These nucleosomes are connected by short stretches of linker DNA, creating a structure that looks like beads on a string. But the packaging doesn't stop there. The beaded string coils into a thicker fiber, and that fiber loops and folds into increasingly complex structures until the DNA is compacted about 10,000-fold. When a cell isn't dividing, this packaged DNA exists in a relatively loose state called chromatin, allowing proteins to access specific genes as needed. But when a cell prepares to divide, the DNA condenses even further into the familiar X-shaped chromosomes visible under a microscope. This packaging isn't just about fitting DNA into a small space. It's also a form of gene regulation. Genes wrapped tightly around histones are generally turned off, inaccessible to the machinery that reads and copies them. Genes in loosely packed regions are more likely to be active. Cells can modify histone proteins by adding small chemical groups, changing how tightly the DNA is wound. These modifications, along with chemical changes to the DNA itself, form the epigenome, a layer of information on top of the genetic code that influences which genes are expressed. You inherit not just your DNA sequence, but also some of these epigenetic marks, patterns of gene activity passed down from your parents. This is one way that environmental experiences can have lasting effects, potentially influencing not just your own development, but also that of your children and grandchildren. The epigenome, a frontier of biology, a realm where nature and nurture intertwine at the molecular level. Humans share most of their DNA with other organisms. It's easy to think of humans as fundamentally different from other species, separate and unique. But at the genetic level, we're remarkably similar to many other organisms. Humans share about 99.9% of their DNA with other humans. The tiny 0.1% difference accounts for all human variation, all the diversity of appearance, personality, and ability that we see across populations. But the similarities extend far beyond our own species. Humans share about 99% of their DNA with chimpanzees, our closest living relatives. We share about 90% with mice, 85% with dogs, and 60% with fruit flies. Even more surprisingly, we share about 50% of our DNA with bananas. This doesn't mean we're half banana. It means that many of the basic biological processes, the fundamental machinery of life, is conserved across vast evolutionary distances. Genes involved in metabolism, cell division, and basic cellular functions are often similar in all living things because they evolved early in the history of life and have been passed down with modifications for billions of years. The genes that make you human, that distinguish you from a chimpanzee or a mouse, are a relatively small fraction of your genome. What makes us unique isn't necessarily having different genes, but using similar genes in different ways, turning them on and off in different patterns, combining them in new sequences during development. Evolution is a tinkerer, working with the same basic parts, rearranging them, modifying them, creating new forms from ancient genetic building blocks. This shared heritage is visible in our DNA. It's a molecular record of our connection to all life on Earth, written in the language of genes that have been copied and passed down through countless generations, adapting and diversifying, but retaining the core sequences that make life possible. DNA can repair itself, but not perfectly. Your DNA is under constant assault. Ultraviolet light from the sun can cause adjacent thymine bases to link together, creating thymine dimers that distort the DNA helix. Free radicals, byproducts of normal metabolism, can damage bases or break the DNA backbone. Environmental toxins can insert themselves between bases or form chemical bonds with DNA. Even the process of DNA replication itself can introduce errors. To counter these threats, cells have multiple DNA repair systems, each specialized for different types of damage. Base excision repair fixes damage to individual bases. Nucleotide excision repair removes bulky lesions like thymine dimers. Mismatch repair corrects errors made during replication. Double-strand break repair fixes the most severe form of damage, when both strands of the DNA helix are severed. These repair systems are constantly at work, scanning your DNA for damage and fixing it before it becomes permanent. It's estimated that each human cell experiences tens of thousands of DNA lesions every day, and the vast majority are repaired without any lasting consequence. But the repair systems aren't perfect. Some damage slips through. Mutations accumulate over time. This is why cancer risk increases with age. It's not just that you're exposed to more carcinogens over a lifetime, it's that your DNA accumulates more and more mutations, and eventually, some of those mutations occur in genes that regulate cell growth and division. There are also genetic disorders caused by defects in DNA repair systems. Xeroderma pigmentosum, for example, is a condition where nucleotide excision repair doesn't work properly. People with this disorder are extremely sensitive to sunlight and have a high risk of skin cancer because thymine dimers caused by UV light aren't being removed. These diseases reveal how essential DNA repair is. Without it, life as we know it couldn't exist. The error rate of DNA replication alone would be too high. Genomes would degrade rapidly, accumulating mutations faster than selection could weed them out. DNA repair is a guardian system, a constant background process that maintains the integrity of the genetic code through countless cell divisions across a lifetime. The genetic code is almost universal. Nearly every living organism on Earth uses the same genetic code. the same correspondence between three base sequences called codons and the amino acids they specify. There are 64 possible three-base combinations using 4 bases, and 61 of these code for amino acids. The remaining 3 are stop signals, telling the cell to end protein synthesis. This universality is strong evidence for common ancestry. All life on Earth descended from a single common ancestor that used this genetic code, and the code has been passed down, virtually unchanged, for billions of years. There are a few exceptions. Some organisms have slight variations in their genetic code. Mitochondria use a slightly different code than nuclear DNA. A few microorganisms have alternative codes, but these are rare variations on the same theme. The near universality of the genetic code has profound implications. It means that a gene from one organism can often function in another. Scientists can take a human gene and insert it into bacteria, and the bacteria will read it using the same genetic code and produce the human protein. This is the basis of biotechnology, the reason we can use bacteria to produce human insulin or yeast to make vaccines. It also means that the boundary between species, which seems so solid and absolute, is somewhat arbitrary at the molecular level. We're all using the same instruction manual, just different pages. The genetic code is also redundant. There are 61 codons specifying only 20 amino acids, which means that multiple codons can code for the same amino acid. This redundancy provides a buffer against mutations. A change in the third position of a codon often doesn't change the amino acid it specifies. These are called silent mutations, changes in the DNA sequence that have no effect on the protein. This wobble in the genetic code, as it's sometimes called, reduces the fraction of mutations that are harmful, giving evolution more room to experiment without breaking essential genes. DNA was once thought to be mostly junk. When the Human Genome Project first sequenced the human genome, scientists made a surprising discovery. Only about 1.5% of the human genome consists of protein-coding sequences, the genes that provide instructions for making proteins. The rest, the vast majority of our DNA, didn't seem to code for anything. This non-coding DNA was often referred to as junk DNA, supposedly useless evolutionary baggage accumulated over millions of years. But this view has changed dramatically. We now know that much of the so-called junk DNA is actually functional. Some of it consists of regulatory sequences that control when and where genes are turned on. Some consists of genes that produce RNA molecules instead of proteins. These RNA genes play important roles in gene regulation, chromatin structure, and other cellular processes. Some non-coding DNA serves structural purposes, providing scaffolding for chromosomes or marking the locations of centromeres and telomeres. It's true that not all of our DNA is functional. Some of it probably is junk, remnants of ancient viral infections or duplicated genes that have become inactive over time. But the proportion of functional, non-coding DNA is much higher than originally thought. The ENCODE project, which aimed to identify all functional elements in the human genome, concluded that about 80% of the genome is biochemically active, though there's debate about how much of this activity is truly functional versus mere biochemical noise. What's clear is that the genome is not simply a collection of genes interspersed with meaningless filler. It's a complex regulatory landscape where genes are just one component. The regions between genes, once dismissed as junk, are now recognized as crucial for orchestrating gene expression, ensuring that the right genes are turned on at the right times in the right cells. This shift in understanding highlights a broader principle. What appears useless or vestigial may simply be poorly understood. The history of biology is full of structures once thought to be vestigial, only to later be found to serve important functions. The appendix was long considered a useless evolutionary remnant, but it turns out to play a role in the immune system. Introns, the non-coding sequences within genes that are spliced out during RNA processing, were thought to be wasteful, but they allow for alternative splicing, increasing the diversity of proteins that a single gene can produce. Even if some DNA is truly junk, it may have been functional in the past, or might become functional in the future. Evolution doesn't produce perfection. It produces organisms that are good enough to survive and reproduce. Carrying some non-functional DNA isn't necessarily a problem, as long as the cost isn't too high. Chromosomes have end caps called telomeres. At the ends of each chromosome are special structures called telomeres. These are stretches of repetitive DNA sequence, thousands of repeats of a 6-base sequence, TTAGGG, that serve as protective caps. Telomeres don't code for any proteins. They don't contain genes. Their purpose is purely structural, protecting the ends of chromosomes from degradation and preventing them from fusing with other chromosomes. Every time a cell divides, its telomeres get a little shorter. This is because DNA polymerase, the enzyme that copies DNA, can't copy all the way to the very end of a linear chromosome. A small segment at the end remains unreplicated. In most adult cells, this means that telomeres shorten with each division. Eventually, after many divisions, the telomeres become critically short, and the cell stops dividing. This is called replicative senescence, and it's thought to be one of the mechanisms of cellular aging. Telomere shortening acts as a kind of molecular clock, counting down the number of times a cell can divide. Some cells, however, don't have this problem. Germ cells, stem cells, and unfortunately cancer cells produce an enzyme called telomerase that adds new DNA sequence to the ends of telomeres, preventing them from shortening. Telomerase essentially resets the cellular clock, allowing these cells to divide indefinitely. In cancer, one of the hallmarks of malignant cells is the reactivation of telomerase, enabling the uncontrolled growth that characterizes tumors. Scientists have long been interested in telomeres and aging. If telomere shortening limits the lifespan of cells, could lengthening telomeres extend lifespan? It's not that simple. While short telomeres are associated with aging, artificially extending telomeres carries the risk of promoting cancer. There's a delicate balance between the need for cell renewal and the need to prevent uncontrolled cell growth. Telomeres sit at this intersection, protecting chromosomes while also limiting the proliferative potential of cells. They're a compromise, an evolutionary solution to the problem of maintaining genome stability while allowing the tissue repair and renewal necessary for a long-lived organism. Every human genome is unique, except for identical twins. No two people have exactly the same DNA sequence. On average, any two humans differ at about 1 in every 1,000 base pairs, or approximately 3 million differences across the entire genome. These differences are what make you genetically unique. They influence everything from your eye color and height, to your susceptibility to certain diseases, and how you metabolize medications. Most genetic variation comes from single nucleotide polymorphisms, or SNPs, places where one person has one base and another person has a different base at the same position in the genome. There are millions of SNPs scattered throughout the human genome. Some are common, found in many people. Others are rare, unique to a small population or even to a family. In addition to SNPs, there are larger structural variations. Entire segments of DNA can be deleted, duplicated, or inverted. These copy number variations can involve thousands or even millions of base pairs and may encompass entire genes. All these variations arise from mutations. Most are inherited from parents, but some are new, arising in the germ cells or early in development. The exception to genetic uniqueness is identical twins. They originate from a single fertilized egg that splits into two embryos early in development. Because they come from the same starting genome, identical twins have the same DNA sequence. But even this isn't entirely true. As twins develop, small mutations accumulate independently in each twin. By adulthood, identical twins have subtle genetic differences, though they're still far more similar to each other than to anyone else. Genetic variation is the raw material for natural selection. It's what allows populations to adapt to changing environments. In a stable environment, most variation is neutral or slightly deleterious. But when conditions change, variants that were previously neutral or even harmful might become advantageous. This standing genetic variation gives populations the flexibility to evolve rapidly in response to new challenges. On a personal level, your unique genome is part of what makes you who you are. But it's not the whole story. Your environment, your experiences, and even random chance all play roles in shaping your development and behavior. Genes provide possibilities, not destinies. Two people with the same genetic variant might have very different outcomes depending on countless other factors. This interplay between genes and environment, between nature and nurture, is one of the most fascinating and complex aspects of biology, a dance between the information encoded in DNA and the world in which that information is expressed. DNA testing can reveal your ancestry. By comparing your DNA to that of people from different populations around the world, scientists can trace your genetic ancestry, revealing where your ancestors likely lived hundreds or even thousands of years ago. This works because different populations have accumulated different patterns of genetic variation over time. People whose ancestors lived in Europe tend to share certain genetic variants that are rare or absent in people whose ancestors lived in Africa or Asia. By looking at which variants you carry, genetic testing can estimate what proportion of your ancestry comes from different regions of the world. Ancestry testing has become increasingly popular, with millions of people sending their DNA to companies that promise to reveal their ethnic heritage. Results can be fascinating, often revealing unexpected connections and migration patterns. Someone who thought they were entirely of European descent might discover they have African or Native American ancestry. Someone might learn that their ancestors migrated from one continent to another, leaving genetic traces of that journey. But these tests have limitations. Ancestry is complex, and the neat regional categories offered by testing companies are often oversimplifications. Human populations have been mixing and migrating for millennia, and genetic variation doesn't respect modern political or ethnic boundaries. Moreover, the accuracy of ancestry estimates depends on the reference populations used for comparison. If certain populations are underrepresented in the database, the test may not be able to distinguish them accurately. Despite these caveats, DNA testing has revolutionized our understanding of human history. By analyzing genetic variation in living people and in ancient DNA extracted from archaeological remains, scientists have reconstructed human migration patterns going back tens of thousands of years. We've learned that modern humans originated in Africa and spread out across the world in multiple waves. We've discovered that humans interbred with Neanderthals and Denisovans, archaic human species that are now extinct, but whose DNA lives on in many people today. We've traced the peopling of the Americas, the Pacific Islands, and every other inhabited region of the globe. Your DNA is a history book, a record of your ancestors' journeys written in the language of genetic variation. Every variant you carry is a molecular artifact, passed down through generations, surviving through countless bottlenecks and migrations, connecting you to people you'll never know in places you may never visit. DNA can be damaged by ultraviolet light. One of the most common sources of DNA damage is ultraviolet radiation from the sun. When UV light strikes DNA, it can cause adjacent thymine bases to link together, forming thymine dimers. These dimers distort the structure of the DNA helix, interfering with replication and transcription. If left unrepaired, they can lead to mutations. This is why sun exposure increases the risk of skin cancer. The ultraviolet light damages DNA in skin cells, and, if that damage occurs in genes that regulate cell growth, it can lead to the uncontrolled proliferation that characterizes cancer. Cells have a specific repair system for dealing with UV damage. Nucleotide excision repair recognizes the distortion caused by thymine dimers and other bulky lesions. It cuts out a short segment of DNA containing the damage and fills in the gap using the undamaged strand as a template. This repair process is highly efficient, fixing most UV damage before it becomes permanent. But in people with xeroderma pigmentosum, a rare genetic disorder, nucleotide excision repair doesn't work properly. As a result, thymine dimers accumulate in the DNA of skin cells. People with this condition are extremely sensitive to sunlight and have a dramatically increased risk of skin cancer. They must take extraordinary precautions to avoid UV exposure, wearing protective clothing and avoiding outdoor activities during the day. Xeroderma pigmentosum is a stark reminder of how essential DNA repair is. We're constantly exposed to DNA-damaging agents, from sunlight to chemicals to the byproducts of our own metabolism. Without robust repair systems, our genomes would quickly accumulate mutations, leading to cell death or cancer. The fact that most of us can spend time in the sun without immediate harmful effects is a testament to the efficiency of our DNA repair machinery, working quietly in the background to maintain the integrity of our genetic information. Even so, it's wise to limit UV exposure. While our repair systems are good, they're not perfect. Over time, unrepaired damage accumulates, and the risk of skin cancer increases with cumulative sun exposure. Sunscreen, protective clothing, and avoiding intense midday sun are all ways to reduce the amount of DNA damage your cells have to deal with, giving your repair systems a better chance of keeping up with the constant barrage of molecular insults. DNA mutations are the basis of evolution. Evolution is often described as descent with modification. The idea that species change over time, with new species arising from ancestral forms. But what drives this change at the molecular level? The answer is mutation. Every new genetic variant begins as a mutation, a change in the DNA sequence. Most mutations are neutral, having no significant effect on the organism. Some are harmful, reducing survival or reproduction. But a small fraction of mutations are beneficial, providing some advantage in the current environment. These beneficial mutations are more likely to be passed on to the next generation, gradually increasing in frequency in the population. Over many generations, the accumulation of beneficial mutations, combined with the elimination of harmful ones through natural selection, can lead to significant changes in a species. New traits emerge, populations adapt to new environments. Over millions of years, this process can result in the diversity of life we see today. The rate of mutation varies across the genome. Some regions mutate more frequently than others due to differences in DNA sequence, chromatin structure, or the activity of repair systems. Mutations also occur through different mechanisms. Point mutations change a single base. Insertions and deletions add or remove segments of DNA. Chromosomal rearrangements move large blocks of DNA to new locations. Each type of mutation has different effects and different evolutionary consequences. One of the surprising discoveries of molecular genetics is that much of evolution may be driven not by changes in genes themselves, but by changes in how genes are regulated. A mutation in a regulatory region can alter when and where a gene is expressed without changing the protein it produces. This can have profound effects on development and physiology. For example, small changes in the timing or location of gene expression during embryonic development can lead to dramatic changes in body structure. This is thought to be how many of the major transitions in animal evolution occurred, not by inventing entirely new genes, but by redeploying existing genes in new contexts. Mutation is random in the sense that it doesn't occur in response to need. A bacterium exposed to an antibiotic doesn't mutate in order to become resistant. Rather, mutations occur constantly, and if one happens to confer resistance, that bacterium will survive and reproduce while others die. This is the essence of natural selection acting on random variation. It's an unguided process, but not a directionless one. Selection filters the variation produced by mutation, preserving what works and discarding what doesn't, gradually shaping populations over time. DNA contains instructions for making proteins, but not all of it does. Genes are often thought of as recipes for proteins, and this is largely true. Most of the genes in your genome provide instructions for building proteins, the large molecules that carry out most of the work in cells. But not all DNA codes for proteins. Some genes produce RNA molecules that have functions of their own without being translated into protein. These RNA genes include transfer RNAs, which help translate the genetic code into protein, and ribosomal RNAs, which form the structural core of ribosomes. More recently, scientists have discovered many other types of functional RNA molecules, including microRNAs that regulate gene expression, and long non-coding RNAs whose functions are still being explored. In addition to genes, the genome contains vast stretches of non-coding DNA. Some of this is regulatory, controlling when and where genes are expressed. Some is structural, providing scaffolding for chromosomes. Some may be truly non-functional, evolutionary remnants that have accumulated over time. The proportion of the genome devoted to each of these functions is a matter of ongoing research and debate. What's clear is that the genome is not simply a linear string of genes. It's a complex landscape with multiple layers of information and function. The physical structure of DNA, how it's packaged and organized in the nucleus, is itself a form of information, influencing which genes can be accessed and expressed. Chemical modifications to DNA and to the histone proteins around which it wraps provide another layer of information, the epigenome, which can be inherited and which influences gene activity. In this view, DNA is not just a storage medium for genetic information. It's an active participant in the cell's life, a molecule whose structure and function are intimately connected. The sequence of bases matters, but so does the three-dimensional organization of chromatin, the chemical modifications that mark active and inactive regions, and the proteins that bind to specific DNA sequences to regulate gene expression. Understanding DNA requires understanding all these levels of organization, from the individual base pairs to the large-scale structure of chromosomes. It's a hierarchy of information, each level building on the one below, creating the complexity necessary to build and maintain a living organism. DNA fingerprinting revolutionized forensic science. In 1984, British geneticist Alec Jeffreys discovered that certain regions of human DNA contain short sequences that are repeated multiple times in tandem. The number of repeats varies from person to person, creating a unique pattern that can be used to identify individuals. This discovery led to the development of DNA fingerprinting, a technique that has transformed forensic science and criminal justice. DNA fingerprinting works by analyzing several of these variable regions in a person's genome. The pattern of repeat lengths across these regions is extremely unlikely to be the same in two unrelated individuals. In fact, the probability of two people having identical DNA profiles is vanishingly small, often less than 1 in a billion. This makes DNA fingerprinting a powerful tool for identification. DNA evidence has been used to convict criminals, exonerate the innocent, identify victims of disasters, and resolve paternity disputes. It has brought closure to families, solved cold cases, and changed the way law enforcement approaches investigations. But DNA fingerprinting also raises important questions about privacy, consent, and the potential for misuse. As more people have their DNA profiles stored in databases, concerns about who has access to this information and how it's used have become increasingly important. DNA evidence is not infallible. Samples can be contaminated, mishandled, or misinterpreted. Lab errors, although rare, can occur, and even a correct DNA match doesn't necessarily prove guilt. It shows that a person's DNA was present at a crime scene, but it doesn't explain how it got there or when. Context and corroborating evidence are still essential. Despite these caveats, DNA fingerprinting remains one of the most powerful forensic tools available. Modern techniques can extract DNA from tiny samples, even from touch evidence or degraded material. Rapid DNA analysis systems can produce results in hours instead of days. And as databases grow, the likelihood of finding matches and solving cases increases. DNA has, in a sense, given us a molecular signature, a unique identifier written into every cell. This signature can be read, stored, and compared, revealing connections and relationships that would otherwise remain hidden. It's a testament to the power of molecular biology to provide answers to questions that once seemed unanswerable, written in the language of the genetic code itself. DNA research led to the biotechnology revolution. Once scientists understood the structure of DNA and how genetic information is stored and expressed, it opened up possibilities that had previously been confined to science fiction. If DNA is universal, if the genetic code is the same in all organisms, then a gene from one species could potentially function in another. This realization led to the development of recombinant DNA technology, the ability to cut and paste genes from one organism into another. In 1973, Herbert Boyer and Stanley Cohen created the first recombinant DNA molecule, inserting a gene from one bacterium into another. This proof of concept launched the biotechnology industry. Scientists could now take a human gene, insert it into bacteria or yeast, and get those microorganisms to produce human proteins. The first major success was insulin. People with diabetes need regular injections of insulin, and for decades, insulin was extracted from the pancreases of pigs and cows. But in 1978, scientists succeeded in inserting the human insulin gene into bacteria. These genetically engineered bacteria produced human insulin, which could be purified and used to treat diabetes. It was a revolution in medicine. For the first time, a human protein could be mass-produced using microorganisms. Since then, many other proteins have been produced this way, from growth hormones to clotting factors to vaccines. Recombinant DNA technology has also enabled the development of genetically modified crops, animals engineered to produce pharmaceuticals, and countless research tools that have advanced our understanding of biology. But genetic engineering is not without controversy. Concerns about safety, ethics, and unintended consequences have accompanied every advance. Genetically modified organisms are subject to extensive testing and regulation, yet debates about their risks and benefits continue. More recently, new technologies like CRISPR have made gene editing even more precise and accessible. CRISPR allows scientists to target specific DNA sequences and make precise changes, opening up possibilities for treating genetic diseases, improving crops, and even potentially editing human embryos. These advances bring enormous potential benefits, but also raise profound ethical questions about how far we should go in manipulating the genetic code. DNA research has not only transformed medicine and agriculture, it has changed our understanding of what it means to be human. We are, in a sense, information-bearing molecules, patterns of DNA that have been copied and passed down through countless generations. Our connection to the rest of life is not just metaphorical. It's written in the DNA we share with other species, a molecular thread linking all living things. The biotechnology revolution is far from over. As our ability to read and write DNA improves, new possibilities will emerge. Synthetic biology aims to design new organisms from scratch. Gene therapy promises to cure genetic diseases by correcting mutations. Personalized medicine tailors treatments based on a patient's individual genetic profile. Each of these fields is built on the foundation of DNA research, on the understanding that life is encoded in a chemical language that can be read, interpreted, and increasingly rewritten. DNA sequencing has become incredibly fast and cheap. When the Human Genome Project set out to sequence the entire human genome in 1990, it was an audacious goal. Sequencing a single gene was painstaking work, requiring months of effort. Sequencing an entire genome, 3 billion base pairs, seemed almost impossible. The project took 13 years and cost about $2.7 billion. But it succeeded, producing the first complete human genome sequence in 2003. In the years since, sequencing technology has improved at a breathtaking pace. Today, a human genome can be sequenced in a matter of days for less than $1,000. This dramatic reduction in cost and time has transformed genomics from a specialized research endeavor into a routine tool used in medicine, agriculture, and countless other fields. Next-generation sequencing technologies work by reading millions of short DNA fragments simultaneously, then using computers to assemble those fragments into a complete genome sequence. The process is highly automated and can be scaled to sequence multiple genomes at once. The implications of cheap, fast DNA sequencing are profound. It's now possible to sequence the genomes of thousands of individuals, comparing them to find genetic variants associated with diseases. Cancer patients can have their tumors sequenced to identify mutations that might be targeted with specific drugs. Newborns can be screened for genetic disorders, allowing for early intervention. Ancient DNA can be extracted from fossils and sequenced, revealing the genomes of extinct species and offering clues about human evolution. As sequencing becomes even cheaper and faster, it will become increasingly integrated into everyday life. Imagine a future where everyone has their genome sequenced at birth, where that information is used to personalize medical care throughout life, where we can sequence pathogens in real time during an outbreak to track their spread and evolution. This future is not far off. It's already beginning in some places. But it also raises questions about privacy, discrimination, and consent. Who should have access to your genetic information? How can we prevent it from being used to deny insurance or employment? What happens if your genome is sequenced without your knowledge or permission? These are not hypothetical concerns. They're real issues that society must grapple with as DNA sequencing becomes ubiquitous. The technology itself is neutral, but how we use it will shape the future. DNA sequencing has given us the ability to read the book of life with unprecedented clarity. What we do with that knowledge will define the next chapter of human history. Some DNA can jump from one location to another. Not all DNA stays in one place. About half of the human genome consists of transposable elements, also known as jumping genes. These are sequences of DNA that can move from one location in the genome to another. Most transposable elements in the human genome are inactive remnants, fossil traces of ancient genetic parasites that inserted themselves into our ancestors' genomes millions of years ago. But some are still active, capable of copying themselves and inserting new copies elsewhere in the genome. Transposable elements were first discovered in corn by Barbara McClintock in the 1940s, though her work was not widely appreciated until decades later. She observed that certain genes seemed to move around the genome, causing changes in pigmentation patterns. It was a radical idea at the time, challenging the notion that genomes were static and unchanging, except for rare mutations. We now know that transposable elements are ubiquitous, found in virtually all organisms. They come in two main types: DNA transposons, which move by cutting themselves out of one location and pasting themselves into another, and retrotransposons, which move by copying themselves into RNA, then reverse transcribing that RNA back into DNA and inserting it at a new location. Most transposable elements are evolutionarily selfish, propagating themselves without providing any benefit to their host. They're genomic parasites, replicating within the genome, much like viruses replicate within cells. Over time, mutations accumulate in these elements, rendering them inactive. The human genome is littered with dead or dying transposable elements, sequences that can no longer jump but remain as molecular fossils. But transposable elements can have important effects. When a transposable element jumps, it can land in the middle of a gene, disrupting its function. It can land near a gene, altering its regulation. It can cause chromosomal rearrangements, deletions, or duplications. Most of these effects are harmful, but occasionally a transposon insertion can be beneficial, creating new regulatory elements or new genes. In fact, some transposable elements have been co-opted by their hosts, turned into functional parts of the genome. Certain immune system genes contain sequences derived from transposable elements. The placenta of mammals uses proteins originally derived from retroviruses. Evolution is opportunistic, taking whatever raw materials are available and putting them to use. Transposable elements remind us that genomes are not carefully designed blueprints. They're more like documents that have been edited, revised, and annotated over millions of years, with layers of text inserted by different hands at different times. Some of those insertions are mere graffiti, meaningless scribbles that happen to have survived. Others have been woven into the narrative, becoming integral to the story being told. DNA can be synthesized artificially. For most of history, working with DNA meant extracting it from living organisms. But scientists can now synthesize DNA from scratch, building sequences base by base in the laboratory. DNA synthesis works by chemically linking nucleotides together in a specified order. Modern DNA synthesizers can produce short sequences, up to a few hundred bases, routinely and inexpensively. Longer sequences can be assembled by stitching together these short pieces. This ability to write DNA, not just read it, has opened up new possibilities. Synthetic DNA is used for countless research applications, from making probes for detecting specific genes to constructing plasmids for cloning. It's used in biotechnology to optimize genes for expression in bacteria or yeast. It's used in medicine to create DNA vaccines, and in agriculture to engineer crops with desirable traits. The ultimate extension of DNA synthesis is the creation of entirely synthetic genomes. In 2010, Craig Venter and his team synthesized the complete genome of a bacterium, over 1 million base pairs, and transplanted it into a cell whose own genome had been removed. The synthetic genome took control of the cell, essentially creating the first organism with a completely man-made genome. This achievement demonstrated that DNA is, in a sense, programmable. We can design sequences on a computer and bring them to life in a cell. It raises the possibility of creating organisms with novel functions, microbes that produce biofuels, clean up pollution, or manufacture pharmaceuticals. But it also raises ethical questions. If we can create life, what responsibilities do we have? What are the risks of releasing synthetic organisms into the environment? How do we ensure that this technology is used for good and not harm? Synthetic biology is still in its infancy, but it represents a fundamental shift in our relationship with DNA. For the first time in history, we're not just reading the genetic code or making small edits. We're writing it ourselves, creating sequences that have never existed in nature. It's a power that comes with profound responsibilities, a reminder that knowledge and capability must be tempered with wisdom and foresight. DNA is no longer just the molecule of heredity. It's becoming a medium for human creativity and design, with all the possibilities and perils that entails. Your DNA is being transcribed right now. At this very moment, inside every cell in your body, DNA is being transcribed into RNA. Transcription is the first step in gene expression, the process by which the information in a gene is used to make a protein. It begins when a protein called RNA polymerase binds to a promoter, a specific DNA sequence upstream of a gene. The polymerase unwinds the DNA double helix, separating the two strands. It then uses one strand as a template, reading the sequence of bases and building a complementary strand of RNA. Unlike DNA, RNA is single-stranded and uses the base uracil instead of thymine. As the RNA polymerase moves along the DNA, it adds nucleotides one by one, creating an RNA molecule that's a copy of the gene. This RNA molecule, called messenger RNA or mRNA, carries the gene's information out of the nucleus and into the cytoplasm, where it will be translated into protein. But transcription is not a constant, uniform process. Cells carefully regulate which genes are transcribed and when. This regulation occurs through transcription factors, proteins that bind to specific DNA sequences and either promote or inhibit transcription. Some genes are expressed all the time, producing the proteins needed for basic cellular functions. These are called housekeeping genes. Other genes are expressed only in specific cell types or at specific times. A neuron expresses genes for neurotransmitter receptors. A muscle cell expresses genes for contractile proteins. The pattern of gene expression defines what a cell is and what it does. This regulation is exquisitely sensitive. Cells respond to signals from their environment, adjusting gene expression in response to nutrients, hormones, and stressors. During development, cascades of gene expression guide the transformation of a single fertilized egg into a complex organism with many different cell types. Errors in gene regulation can have serious consequences. If a gene that's supposed to be turned off is instead expressed, it can disrupt normal cellular function. This is one way that cancer can arise. A gene that promotes cell division might be turned on inappropriately, leading to uncontrolled growth. Conversely, if a gene that's supposed to be expressed is silenced, essential proteins may not be made, leading to disease. Transcription is not just about copying DNA into RNA. It's about controlling that process, ensuring that the right genes are expressed at the right times in the right amounts. It's a dance between DNA, RNA, and proteins, choreographed by evolution to build and maintain life. DNA can form structures other than the double helix. While the double helix is the most famous and most common structure of DNA, it's not the only one. Under certain conditions, DNA can adopt alternative structures with different properties and functions. One such structure is the Z-DNA, where the helix twists in the opposite direction from the standard B-DNA. Z-DNA forms in regions with alternating purine and pyrimidine bases, and is thought to play a role in gene regulation. Another structure is the triple helix, where a third strand of DNA winds into the major groove of a double helix. Triple helices can form naturally in certain DNA sequences, and can also be created artificially to target specific genes. DNA can also form quadruplex structures, where 4 strands come together, stabilized by unusual base pairing. These G quadruplexes form in regions rich in guanine and are found at the ends of chromosomes, in telomeres, and in certain gene promoters. They're thought to play roles in telomere maintenance, gene regulation, and genome stability. Under stress, DNA can become supercoiled, wound tighter or looser than the relaxed double helix. Supercoiling is important for packaging DNA into chromosomes and for regulating access to genes. Enzymes called topoisomerases manage supercoiling, cutting DNA strands, allowing them to rotate, then resealing them. These alternative structures are more than curiosities. They're functional elements of genome biology, involved in replication, transcription, and chromatin organization. They show that DNA is not a static, rigid molecule, but a dynamic one, capable of adopting different shapes in response to cellular needs. The flexibility of DNA, its ability to bend, twist, and form loops, is essential for its function. Proteins bind to DNA not just by recognizing specific base sequences, but also by sensing the shape and flexibility of the helix. Some proteins bind preferentially to bent DNA. Others induce bends when they bind. This interplay between DNA structure and protein binding adds another layer of information to the genome, beyond the simple sequence of bases. Understanding DNA requires understanding its structure at multiple levels: the chemical structure of bases and the backbone, the double helix, higher-order structures like quadruplexes and supercoils, and the packaging of DNA into chromatin and chromosomes. Each level contributes to the overall function of the genome, creating a hierarchy of organization that allows information to be stored, accessed, and regulated with exquisite precision. DNA is not just information, it's also architecture, a molecule whose shape is inseparable from its function, a physical structure that embodies the logic of life. DNA methylation can silence genes without changing the sequence. One of the most important epigenetic modifications is DNA methylation, the addition of a methyl group to certain bases in DNA, usually cytosines. Methylation can change how a gene is expressed without altering the underlying DNA sequence. When a gene's promoter region is methylated, it's typically turned off, silenced. Methylation patterns are established during development and are maintained as cells divide. They're one way that cells remember what type of cell they are. A neuron keeps neuron genes active and liver genes silent, in part through methylation. But methylation isn't permanent. It can be added or removed in response to environmental signals, providing a way for experience to influence gene expression. For example, studies in rats have shown that maternal care affects methylation of genes involved in stress response. Pups that receive high levels of maternal care have different methylation patterns than those that don't. And these differences persist into adulthood, affecting how the animals respond to stress. Similar effects have been observed in humans, suggesting that early life experiences can leave lasting molecular marks. But methylation can also go awry. In cancer, genes that normally suppress tumor growth can become hypermethylated and silenced, contributing to uncontrolled cell proliferation. Conversely, genes that are normally methylated and silent can become demethylated and active, leading to inappropriate gene expression. DNA methylation is just one of many epigenetic modifications. Others include modifications to histone proteins, changes in chromatin structure, and regulation by non-coding RNAs. Together, these mechanisms create a layer of information on top of the genetic code, influencing gene expression in ways that are dynamic, responsive, and potentially heritable. The discovery of epigenetics has changed how we think about heredity. It's not just about the sequence of bases in DNA. It's also about the chemical modifications and structural changes that affect how that sequence is read. Identical twins, who start with the same DNA sequence, can end up with different epigenetic marks due to different life experiences, leading to differences in gene expression and phenotype. This explains why identical twins aren't truly identical, why they can have different susceptibilities to disease and different personalities. Epigenetics also offers hope for treating diseases. If gene expression can be altered by changing methylation patterns or histone modifications, then drugs that target epigenetic mechanisms might be able to turn genes on or off therapeutically. Some such drugs are already in use for treating certain cancers. It's an active area of research, with the potential to develop new therapies for a wide range of conditions. DNA sequence is destiny, but it's not the whole story. Epigenetics adds context, flexibility, and responsiveness, allowing the same genetic information to be interpreted in different ways, depending on the cellular and environmental context. It's a reminder that biology is not just about the information in our genes. It's about how that information is used, regulated, and modified in the complex dance of life. DNA can be edited with precision using CRISPR. In 2012, scientists Jennifer Doudna and Emmanuel Charpentier discovered a bacterial immune system that could be repurposed as a powerful gene editing tool. This system, called CRISPR-Cas9, allows researchers to target specific DNA sequences and make precise changes, cutting, deleting, or inserting genetic material with unprecedented ease and accuracy. CRISPR works by using a guide RNA that matches the target DNA sequence. The Cas9 protein, guided by this RNA, finds the matching sequence in the genome and cuts both strands of the DNA. The cell's own repair systems then fix the break, and during that repair, scientists can introduce changes, either knocking out a gene or inserting a new sequence. The simplicity and versatility of CRISPR have made it a revolutionary tool in biology. Researchers use it to study gene function, to create animal models of human diseases, to develop new therapies, and to improve crops. In medicine, CRISPR holds the promise of curing genetic diseases by correcting the mutations that cause them. Clinical trials are underway for treating conditions like sickle cell disease and certain forms of blindness. But CRISPR also raises ethical questions, especially when it comes to editing human embryos. In 2018, a Chinese scientist announced that he had used CRISPR to edit the genomes of twin girls, making them resistant to HIV infection. The announcement was met with widespread condemnation from the scientific community. Editing embryos means making changes that will be passed on to future generations, changes whose long-term effects are unknown. Most scientists agree that germline editing, altering the DNA of embryos or germ cells, should be subject to strict ethical oversight and should only be considered for serious diseases with no other treatment options. The technology is still new, and its full implications are only beginning to be understood. CRISPR is more than just a tool. It represents a fundamental shift in our relationship with DNA. For the first time, we have the ability to edit the genetic code quickly, cheaply, and precisely. This power comes with responsibility. How we choose to use it will shape the future of medicine, agriculture, and perhaps even human evolution itself. DNA is no longer just something we inherit and pass on. It's something we can modify, for better or worse, with consequences that will echo through generations. And so, let the facts settle gently around you like a soft blanket. You've drifted through the spiral of DNA, through its discovery and structure, through its function and its mysteries. You've learned about the codes that build proteins, the mutations that drive evolution, the repairs that keep our genomes intact. You've explored the packaging of chromosomes, the dance of replication, the promise and peril of genetic engineering. All of this exists within you, right now, in every cell. Your DNA is at work, transcribing genes, replicating itself, repairing damage, maintaining the intricate machinery of life. It does this without your conscious thought, a quiet symphony of molecular interactions that began billions of years ago and continues in you, in me, in every living thing. Tonight, as you drift towards sleep, know that you are part of an ancient lineage, a chain of life stretching back to the very beginning. The DNA in your cells is a gift from your parents, and from theirs, and from theirs, an unbroken thread connecting you to the first organisms that lived on this planet. You are made of stardust and time, of molecules that have been recycled through countless generations, shaped by evolution into something remarkable. You. Rest now. Let go of the day. Let your breathing deepen and your mind quiet. The universe will keep turning without you for a while. Your DNA will keep working, faithfully copying itself, expressing genes, maintaining order in the face of entropy. You have done enough for today. Close your eyes, rest, and dream. Good night.